I'm Dr. Chelsea Himsworth, the lead researcher for the Vancouver Rat Project. Although rats are an important source of disease for people living in cities around the world, we know remarkably little about them, particularly here in Canada. By trapping and sampling rats collected throughout Vancouver's downtown east side, we hope to determine what infectious agents rats carry and how these agents circulate in rat populations. This information will help us to gauge the health risks posed by urban rats and to develop strategies to monitor and mitigate those risks. One of the disease agents we looked for is called Leptospira. It's a spiral-shaped bacterium that is carried in the rat kidney and shed in the urine. Although Leptospira is harmless in rats, infection in people can lead to fever, kidney failure, and even hemorrhage into the lungs. We found that many rats in the downtown east side were infected with Leptospira, but that the rate of infection varied remarkably, between 0 and 66% among the different city blocks. Urban rats live in tight colonies, with small home territories often less than 50 meters in diameter. Colonies are effectively isolated from their neighbors by physical barriers like roads and by social pressures from adjacent rat colonies. This means that while one block may have a lot of Leptospira-carrying rats, the adjacent block might have none. We're still trying to figure out why some blocks have Leptospira-carrying rats and others don't. We also found that older rats, fatter rats, and rats with more bite wounds were more likely to carry disease. This is interesting because we used to think that rats became infected with Leptospira through random exposure in the environment. But age, body fat, and bite wounds are all factors associated with dominance and social hierarchies, suggesting that Leptospira is probably transmitted through social contact among rats. This has implications for our current methods of managing rat populations through trapping and poisoning. Studies in other colony-forming species have shown that killing a few individuals can destabilize the family unit, causing migration of infected animals to uninfected populations and increasing fighting behavior, both of which can facilitate disease spread. This means that traditional trapping and poisoning might actually increase the number of rats infected with Leptospira, and this is something that will need to be investigated in future studies.